Okay, I'm trying to connect to YouTube before I start the class. I want to believe they have joined. I want to believe my YouTube is up. Okay. I can see that one is up. Okay. I'm sure you can hear me. Good morning, everybody. I thought you said we should drop. You are hearing a miss. Eh? Beautiful and Otun. You are hearing a miss. All right. Good morning, everybody. Please let the attendance stop coming. Let the attendance stop coming so that we can all enjoy the class for today. So good morning once again. I'll be standing in for Dr. Pao, who is unavoidably absent, and we'll be finishing his part today by God's grace. On Monday, another lecturer will come in. You still have 3 more topics to cover by 3 different lecturers. So we still have a long way, although we have covered more than 50%. By the end of this class, we will have covered about 4/7 of the lecture of the topics that we are supposed to take for the course this session. And you guys are preparing for your tests. As you are doing Valentine, remember that BIO101 test is in a fortnight, and do not Valentine away your grades. And always remember to read your notes. If you have not started reading, take your notes now and start studying. The test will cover the topics that we have treated so far, starting from scientific theory and what have you, my first class, then to biomolecules and central dogma, to cell structure and function by Dr. Adebeshin, and this topic that we are rounding up today. These are the 4 areas that will be treated or will be touched in your first continuous assessment test, which we hold by God's grace on February 28th. That is 2 weeks' time. And the second test, the dates will be announced after the first test. Then we cover what you are going to do subsequently, the remaining 3 topics. That will be reproduction and development, then ecology by Dr. Dada, and the classification and evolution of living things by Professor Bunkami. Those 3 topics we make up your second test. The first test we hold on Friday, and there is going to be the 4 topics that we have covered so far. Then I think we have passed that information concerning lab, grouping, deadline for registration, and what have you. Then before we start our lab, I would like to have a live session with everybody so that we can know what is expected of us during the practical, because you have a very short time for practical, right? So I want to tell you what we expect of you so that when you come, you don't run foul of any of the rules for that practical, so that you can come in, do your lab in the shortest possible time and leave. Submission and all of that will be treated maybe a day to starting practical. Somebody asked if the test will be physical. I'm not bringing you from your house to come and write test. I think we have said the modality will be announced. So please, the only thing you should bother yourself about now is reading, studying. The logistics, leave that to us. We will provide information as and when due. So don't bother yourself how the test we hold. It's going to be online, and the other things will be provided as and when due. Just go and be studying. What you want to write is what matters, right? Okay. Good morning once again, everybody. Let's start the class. Dr. Shifao, like I said earlier, is unavoidably absent, and I think this is where he stopped with you the last time. Maybe as a recap, if I know what he taught you people. You must have heard of Gregor Mendel, you must have heard of crosses, you must have heard of the young man, please, nobody should drop anything in my chat. In fact, let me even mute this chat room. Let me off the notification. You didn't send a slide on cell reproduction. Which one is cell reproduction? Cellular reproduction and heredity. That is Dr. Shifa's class. Is the one that you will ask for that slide. And I think I sent it. I sent the one that I took, the part that I took for him. Meiosis and mitosis. I'm sure I sent that part. So any slide that you'll be asking is this genetics, and there's no way we are sending the slide when the class has not ended. So expect this one subject to his approval when his class is over. All the lectures that we've had in the past, we've shared the slides. So if you can't find them, ask your course rep. I mean, those that send notification to your platform for lectures. Ask them, even including the recordings of these lectures. We keep all of them and we share with your reps. So please, all the lectures that you've had so far, we've sent the slides to you. Okay. Okay, so I was talking about genetics. The term genetics was coined by William Bateson in 1806, if I'm correct, or I think it's 1806, not 1906. I think it's 1806. Then Gregor Mendel, an Austrian monk, pioneered the field of genetics using garden pea sativum, but his work was neglected for 34 years or thereabout, about 40 years. Nobody thought it was making sense until three scientists, if I remember their name now, Gouffers, Chemark, and De Vries, now rediscovered his work while working on ABO block group and all of that. And his work now gained a prominence, and he laid the foundation for what we know in genetics today. That is our Father in the Lord in heaven, Gregor Mendel, of blessed memory. May the Lord continue to be with his soul. Okay, so then there are some basic terms that you are supposed to familiarize yourself with in genetics, which I'm sure Dr. Shifa must have dealt with. So we are not going to recap what he has done. So today we're going to continue with the Mendelian laws of inheritance and look at some crosses, genetic crosses to buttress our points on these laws. So we have the law of segregation, which states that during gamete formation, the alleles for each gene pair segregates such a way that gametes carry only one allele for each gene. Please hold on. Okay, sorry, I'm back. So we have the law of segregation, which says that during the gamete formation, gametogenesis, there is formation of gametes, and we have our gametic cells, which are those cells that are involved in reproductions, right? So we have genotypes, which are genetic combination, right, that determines phenotype, physical appearance. So when gametes are formed, right, for instance, let's look at the traits, tallness, height rather, in plants. Let's assume that a plant is heterozygous tall. Now, the law of segregation is saying that during gamete formation, that is when you are making your sperm cell in males and your egg cells in females, these genotypes, these are alleles, right? These alleles that make up the genotype. You have the allele capital T for tallness and small letter T for dwarfism. So during gamete formation, this genotype, right, TT, will segregate so that each gamete, whether egg cell or sperm cell now, we only have only one of these alleles. That is, a sperm cell can only carry capital T or small t. That is, they will separate, they will segregate. You know, they are together, they pair up to determine a particular trait, right? So during gamete formation, these alleles will separate so that an egg cell can only carry this or this. That is for heterozygous genotype. But when you have a homozygous, as in maybe homozygous dominant or homozygous recessive, what can you have here? You can only have for homozygous, you can only have a particular type of allele because the alleles are similar. They are both dominant or recessive. So in this type of genotype, you can only have dominant allele. In recessive, homozygous recessive, you can only have a recessive allele. So that is why the law of segregation is saying segregation, separation. The genotype will break apart into their respective alleles, and the gametes can only carry one type of allele, not two alleles now going into a gamete. Then you have the law of independent assortment. Briefly, what the law of independent assortment is saying that when you have more than one trait under consideration, when you have two traits that are determined by two genes, right? The inheritance of one trait is independent of the other traits, or the alleles are independent. So for instance, you have two traits, maybe flower color, purple, and the plant height, tall. Now, what the law is saying is that, so let's assume these alleles are the traits are heterozygous. Maybe we have capital P small p, capital T small t. What the law of independent assortment is saying is that here you can have this capital P small p breaks down into one capital, one small. This big T small t, right? So what the law of independent assortment is saying is that where this capital P will go in a gamete is independent of where the small p will go, or the capital T. So it doesn't mean that once you have a dominant allele for a particular trait, then you are going to have the other one too must be a dominant allele or recessive. So it doesn't matter. They are independent. Not that when you have a recessive allele for one trait in one gamete, then the same gametes will also have recessive traits for the other traits. Recessive allele for the other traits, no. So that is what the law of independent assortment is all about. I'm sure Dr. Shifao has done justice to this, and just want to, because this will form the basis of what we are going moving forward. Then the law of dominance is generally not regarded as law because it has been disproven by, there are some traits that not obey law of dominance. That is, the dominant allele is not even dominant at all, or it's not completely dominant, as we shall see under exception to Mendelian principle. So that is all about the Mendelian law of inheritance. So let's look at monohybrid cross to illustrate, to drive home our points on the law of segregation. Monohybrid simply means parents that differ by a single trait, that is, inheritance of single traits. Mono, one, hybrid, crossing individuals, right? So monohybrid cross is talking about inheriting a particular trait at a time, one trait per time. So crossing two pea plants that differ in pod color. The pod color here is our trait under consideration. And don't forget, for a particular trait, you have two alleles according to Gregor Mendel. for a particular trait, there's only two alleles and is determined by only one gene. That is recommended. There are exemptions. Okay, so for pod color, we have two alleles. And Dr. Shifa must have mentioned that recommended studied 7 traits in Pisum sativum. Pod color, seed shape, seed color, what was it called? Plant height, flower position, flower color, and what have you, right? You can have red, round, and wrinkle, right? Okay, so in this, for pod color, you have one allele for green color and the other allele for yellow color. And the green here is dominant over the yellow allele. That is for pod color. But for seed color, as we shall see, the yellow is dominant over the green. That is for seed color. Okay, so what Gregor Mendel observed was that when he crossed a true breeding green pod with a true breeding yellow pod, all the offspring F1 generation are green. Now, you are hearing true breeding. When we say true breeding in genetics, true breeding or pure breeding, pure, P-U-R-E, or true breeding, it means that the genotypes are homozygotes for the trait under consideration. For instance, if for a true breeding green, it means that that green is homozygous green. You know that for a dominant trait, the trait will be expressed whether as homozygotes or heterozygotes. That is, whether this plant is capital G, small g, heterozygous, or capital G, is going to be green as long as you have your dominant allele G, right? So when you hear true breeding, it means the plants under consideration or the organism. It could be coat color, inheritance in rabbit or what have you, right? It means that the trait under the organism is homozygous. If it is green, it is homozygous green. If it is yellow, definitely yellow is a recessive trait, it has to be homozygous recessive. Now, when Gregor Mendel crossed these true breeding green plants, pod color, and true breeding yellow color, he observed that all the offspring are heterozygous dominant. They are heterozygous dominants here, right? You see this green crossed with yellow. This is the F1, all green. But you will agree with me that they are going to be heterozygous green, right? So let's quickly, okay, let me see whether the next slide covers, shows the crossing and how you arrive at heterozygous green. Okay, no. So let's quickly look at how this green, homozygous, true breeding green, and true breeding yellow. Don't forget, for the recessive trait, you don't use Y for yellow, right? It is the same letter that you use for the dominant that you use for recessive, but you now write this in a lowercase. Now, this is homozygous green and homozygous yellow, and all the F1 generation are heterozygous green. So let's look at the crossing. According to Mendelian genetics, to the law of segregation, right, that we are looking at, we have homozygous green and homozygous yellow, right? So during gamete formation, this is the parental genotype, right? So during gamete formation, you are going to have only one particular of allele from here, or gamete, that is capital letter G. Then this one will be small letter G. In genetics, we put gamete in cycle to differentiate it from any other, maybe from allele, right? This is a gamete in cycle. So when you cross this, you will agree with me that you can only have capital G, small g in all of the 4 individuals. And these guys will be green, but heterozygous green. So that is why you are saying that your true breeding green and true breeding yellow gives all green. But these green are heterozygous. They are not homozygous like the parents, like the parental genotype. Okay, so Gregor Mendel was now wondering that, how can I cross green and yellow will disappear? Where did my yellow go to? Where is my yellow pod color? And I said, okay, I know what I will do. Let me cross this F1. Let me self them. Selfing. Selfing is when you cross individual with itself. that is the same genotype. If that individual is heterozygous, you are going to cross with an heterozygous. If it is homozygous, you are going to cross with an homozygous. So that is called selfing. You are selfing, like when you go to your bank to withdraw your money yourself, right? You are selfing that individual organism or plant in most cases. We do experiments with plants. You don't self human beings. Okay. So Gregor Mendel now self F1, that is self-fertilizing F1. So if you let F1 generation to self-fertilize, the next monohybrid cross will be, we now have our Punnett square to illustrate what F2 will look like. So from our F1, we said that all our F1 are heterozygotes, right? So they will form gametes. This is gamete for one parent, F1 parent. Then this is another gamete for one F1 green. So when you cross this dominant G and dominant G, you have what we call here, that's 1. When you cross dominant G and recessive G, you have 2. When we cross dominant G and this, you have 3. When you cross the 2 recessives, you have 4. So this is Punnett square. Now, if we now observe this clearly, we will see that we have genotypes 1, homozygous green, homozygous, that will be GG1. This is that. Then for the second genotype that we have heterozygous, that is 1 and 2. Then the third form of genotype that we have is the recessive, which is small letter g, small letter g. So that means we have 3 possible genotypes: 1 homozygous green, 2 heterozygous, and 1 homozygous recessive. And that is why you have our genotypic ratio, that is the ratio of the different genotypes that we can have from a monohybrid cross, all things being equal, according to Gregor Mendel, is 1:1. But for phenotypic ratio, the phenotype, the physical appearance that you will see is that you will see green and yellow. And the green will be those homozygous and heterozygous, 1 + 2, 3. And the recessive allele, yellow. So for phenotypic ratio is 3:1 for a monohybrid cross, and the genotypic ratio is 1:1. Okay, I muted the chat room because of this. Okay, there's no message here. There's no message here, except attendance that I don't like seeing. Okay. So that is for monohybrid inheritance. As a recap, we said monohybrid crossing means inheritance of one trait, and the Gregor Mendel studied that by crossing pure breeding individuals, pure breeding for green and pure breeding for yellow, right? We can also have pure breeding for tall and dwarfism for short, or dwarfism or dwarf. That means homozygous tall and homozygous short. Okay. So after that, across them, he realized that all the F1 generation heterozygous green, it now did selfing. When self-fertilized, it now said that, okay, my yellow pod is back here. And you have your genotypic ratio as 1:1 and phenotypic ratio as 3:1 for a monohybrid cross. So let's look at dihybrid cross, or the law of independent assortment, which means that members of one GPS segregate independently from other gene during gamete formation. And then let's look at that law with a dihybrid cross, right? Because we're looking at independent assortment now. We're looking at traits that are more than 1, could be 2, 3, 4, 5, 6, right? So at a time, so we are going to look at that using 2 different traits. Dihybrid, 2 inheritance. That's inheritance of 2 traits. You can have trihybrid, you can have tetrahybrid, but we are going to stop at trihybrid for this class. Sorry, at dihybrid rather for this class. So we have crossing in plants that are true breeding for 2 different traits. Like I said earlier, true breeding and pure breeding means homozygous genotype. So if you are breeding for pure breeding tall, that will be 2 capital T. Pure breeding short, small letter t. Okay, now the two traits that we are looking here are pod color and seed color. And don't forget, these traits are inherited the opposite way in Pisum sativum. For instance, pod color, the dominant is green. Seed color, the dominant is yellow. Pod color, the recessive is yellow. Seed color, the recessive is green. Okay, so let's look at this slide on the screen. We have true breeding green with yellow seed. That is, the pod is green, but when you check inside, the seed inside the pod is yellow. The other one, the pod is yellow, but if you check inside, the seed inside the yellow pod is green. These are two different traits. Pod color, one trait on its own. Seed color, other trait on its own. And I said that for pod color, the dominant trait is green, and the dominant seed color is yellow. The dominant, the recessive pod shape is yellow, and the recessive seed color is green. So when you cross that, don't forget we're looking at two traits. That's why we have two genotypes here. So we have, for the true breeding green pod with yellow seed, you have this genotype. That is green, green, yellow, green and yellow. Then for the true breeding yellow pod with green seed, you have this genotype. Now you agree with me that when you want to form gametes from this true breeding green pod yellow seed, you can only have, for the green, let's take it one by one, you can only have capital G as your gamete, right? Then for the Y, you can only have capital Y. So that is why you are having gamete is equal to capital G, capital Y. Then for the other traits that has recessive genotypes, GG and YY. The gamete that you can form from this GG is recessive G, and the gamete you can form from this YY is a recessive Y. So that is why for the other true breeding recessive traits, you have small letter G, small letter Y. For this one, you have capital letter G, capital letter Y. Now, when you cross these two guys, you will agree with me that this is what you are going to get in all the 4 individuals. You are going to get heterozygous green and heterozygous yellow, like we got for monohybrid inheritance. So if you don't understand how that, just pick the traits one by one. When you cross GG, GG, you are going to get heterozygous G, yes or yes. Then when you cross YY, recessive Y, the only offspring you are going to have here is capital and small, dominant heterozygous yellow. So that is why all the 4 offspring, like our F1 generation, all the 4 offspring, we have this genotype. That is, they are heterozygous green pod with yellow seed. Heterozygous green pod with yellow seed, all the F1 generation. Like we did for monohybrid, let's do again for dihybrid. And what did Gregor Mendel do is by selfing. When you now self your F1, when you self your F1, you will agree with me that the only thing you can get here is when you self, by selfing F1, we mean we are crossing this with this, selfing the same genotype. So for this, you are going to have the same gametes. So here, you are going to form this because they are the same genotype. So you are going to have the same gametes in both the male and the female plants. Let's see whether we have any concern on YouTube before we proceed. Or let me first check the chat room here that I've muted. Okay, some people are still dropping attendance still. Now, what for now? People do not do well in my exam. I'm going to take a screenshot of where I drop attendance here. If you don't do well in the test, you see what I will do for you. I'm going to beat all of you. I don't know. Simple instruction. Better read the instruction before you write your tests. Ability to read and follow instruction is part of examination. People don't follow instruction. Okay. Okay, let's see what is happening on YouTube. Maybe all is well with our gentlemen and ladies over there, or they have gone on Valentine. YouTube family, are you there? If you can hear me on YouTube, say hi. YouTube. YouTube, let me see your message now. No, sir, it's clear. What of sales link trades? What is your business with sales link trades? Hi, thank you, Jare. Thank you. All is not well, sir. What happened? Hi. If all is not well, you can enter my DM and ask any question, because YouTube is the message keep coming and we may not be able to follow through. So, like I always say, you have I see somebody sent a message as late as around 9 or 10 yesterday PM. So you have our contact number, you have my email, you have my phone number. You can come to my DM. Colleague said yesterday, when you type a message, good morning, sir, I am so and so from so and so department. Please, I need clarification on so and so. Then I will reply your message, and please don't use timer for your message when you are sending me a message on WhatsApp, because I may not be able to reply your message within 24 hours. And if you have used 24 hours timer for your message, sorry. Okay. So, how do I get your contact, sir? If you can hear me, that person that asked that question on Facebook, my phone number is 070-3556-9436. 070-3556-9436, or send an email to tfakorede together, no dot, no space, tfakorede, T for Taiwo, tfakorede@unilag.edu.ng. If you ask for that on Facebook, on YouTube, please let's. Okay, I hope this question stays. Sir, what if two different alleles that are dominant cross, like homozygous red flower and homozygous yellow? What will be the color of the F1 generation? Homozygous what? Homozygous red flower and homozygous yellow. Homozygous yellow and flower color in plants, you can't have two homozygous for the same part for a particular trait. For instance, now in the particular flower, the flower color is homozygous yellow. Then that particular plant, the dominant trait there, homozygous, is black or green. No, it's not possible. Flower color is a trait. So you can't have two traits for flower color. So you can now have different characters. You can now have, for a flower color as a trait, you can have the character as maybe green flower color or yellow flower color. But the one recommended study were purple and white. Purple is dominant over white. I think you get that explanation. You cannot have, for instance, now for plant height, you're having tall as dominant, then you are having semi-tall as dominant. I don't know what to call that. You cannot have two homozygous phenotypes for a particular trait. It's not possible. So that's your question, my brother. Don't confuse yourself well. Okay, so let's move on. Let's move on. So when we now cross these two, when we self F1 for our dihybrid, that is, we're crossing this on the screen. Now we need to form gametes, and you will agree with me that we can only form the same gamete for both of them because the genotypes are the same. So let's form our gamete. The gametes are here, right? But how do we form these gametes? What if I don't use GG? What if I use A and B? And I ask you to give me the possible gametes that can be formed from this genotype. So let's quickly look at that trick. I will take it once. So please follow me. So let's assume we have this genotype to form a gamete for. So we can use what we know as the four-line rule. The four-line rule goes this thus. You have your G for the second allele, dominant G. Then the next one is small letter G. You write it under it. Then you spread the next allele on them. So you have capital Y, small y. You have capital Y, small y. All right. So let me erase it and do it. We want to form gametes for this. I said using a four-line rule, you can do it this way. You pick the first allele, capital G, the second allele, small g. Then you spread the next alleles on each of them. The next one is this and this. The next one is this and this. Even if you have AA, maybe you're doing trihybrid, you spread your AA on this, right? You spread your AA on them like this. So that is how to determine gametes for dihybrid, and when you're looking at more than one, what's it called? More than one traits. So here we have this. So how do we now form our gametes? Let's use yellow color to form our gametes. We come here. The first one is capital G, capital Y. That is capital G, capital Y. That is one gamete. The next one is to go under capital G, small y. The next one, small g, capital Y. And the last one, small g, small y. So that is how to form gametes for monohybrid, for heterozygous genotypes. When you have more than one trait, when you have more than one trait. So, and you can determine that by using a formula. The number of different gametes that you can have for heterozygous genotypes is 2 raised to power n, where n equals number of heterozygous genotypes. Number of heterozygous genotypes. Now, how many heterozygous genotypes do we have here? 2, right? So how many gametes can we form? Is 2 raised to power 2, which is 4, like we see here. This is our heterozygous genotype, is 2, right? This is one heterozygous. Capital G, small g is one heterozygous. Capital Y, small y is another heterozygous. So how many gametes can we form from this? That is 2 raised to power n. That is 4. Now, relying on this, I need you to tell me how many gametes we can form for this particular genotype. Can we form for this particular genotype using 2 raised to power n? Look at the genotype here on the screen. We have BB, CC, DD. That is heterozygous B, homozygous C, and heterozygous D. We now have 3 traits. How many type of gametes can we form here? I need your answer in the chat room now. Those of you that like writing attendance, oh yeah, write your answer. 8, 6. These are the options in the exam. A, 4, B, 6, C8. Nobody said 12. 4, 4, 6, 8. Okay, none of the above. Option D will be all of the above. Okay, okay, okay. The correct answer is 4. The correct answer for this is 4, because we said N equals number of heterozygous genotype. And how many heterozygous genotype do we have here? Although we have 3 traits, but there are only 2 heterozygous genotypes, which is B, capital B small b, and capital D small d. This one is not taken into that formula because it's not heterozygous. This is homozygous. It can either be homozygous dominant or homozygous recessive. But as long as they are homozygous, they are not taken into that formula that I gave you. That is how many gametes you can form in dihybrid and more than one traits. That's the number of genotype you can form is 2 raised to power n, where n equals number of heterozygous genotypes. And how they will look like, I've shown you how to do your 4 clients to get those 4 that will form for that. Okay, without wasting time, our time is fast spent and we still have a long way to cover. I might spend like 15 minutes extra. And I'm begging. I hope you don't have any class by 9. If you have any class by 9, you can drop it in the chat room. Let's see, just answer any class by 9. We do. Who are we? Sir, we do. Who are we? Who are we? Who are we? We have, we do. Malo class. Mosibo, you can go for your class. HN101 by 9. No problem. Go for your class. I won't take your time. So I will not be spending my time to explain again. I will just explain briefly. Okay. So we have this, selfing F1 for a dihybrid. So we have these genotypes, these gametes that are formed. And you agree with me that since they are the same genotype, they will have the same gametes. And these gametes are here on the horizontal and the vertical axes. And when you cross them, I think you must have done this in your secondary school biology, if you attended a good secondary school like me. Yes. Okay. So we have this and this. When you cross capital G, capital Y, let's use dominant. Just be fast with it. When you cross dominant G and Y with dominant G and Y, you have this. That is heterozygous, homozygous green pod with yellow seed, as you can see there. So if you do that correctly, you are going to have 4 different phenotypes, of which 9 are green pod and yellow seed. Let's quickly count those ones with green pod and yellow seed. 1, green pod yellow, see green and yellow, 2, 3, 4, 5, 6, 7, 8, and 9. Those ones are green pod and yellow seed. Then you have 3 that are green pod and green seed. Green pod and green seed. You see 3, 1, 2, 3. Then you see another 3 that are yellow pod and yellow seed. Yellow pod and yellow seed. 1, 2, 3. And the homozygous recessive for the 2 of them that are yellow pod and green seed. Yellow pod and green seed. So the phenotypic ratio for a dihybrid cross is 9:3:1. The phenotypic ratio now is 9:3:1, whereas for monohybrid is 3:1. So from this crossing, you can now have 9 different genotypes, making up 4 phenotypes. And the genotypes and phenotypes are as shown on the screen. So let's look at Mendelian rules. Mendelian rules say that all genes have 2 allelic forms, one dominant and the other one is recessive. All traits are monogenic, that is, they have only one gene in a locus. All chromosomes occur in homologous pairs, like we know, homologous pair chromosome 1 to 22, then your sex gametes. All genes assort independently according to the law of independent assortment. Then a mutation in a single gene can cause a disease that is inherited according to Mendelian laws. and you have known I've treated mutation with you. Then our class on cell on a DNA replication, how DNA mutation can cause hemoglobin normal to hemoglobin sickled. So, and the examples of these traits that follow Mendelian principle are sickle cell, Tay disease, cystic fibrosis, and xeroderma pigmentosa. All of these obey Mendelian principle. That is, they have one gene. The gene has two alleles, one dominant, one recessive, and as you can have them. So let's quickly look at non-Mendelian inheritance. That is, what are these? Don't forget when we said we have three laws of inheritance. Mendel prepared three laws, but the other one, the law of dominance, is disproven. So these ones do not obey Mendelian principle. Mendel said that all traits have only one gene, and those genes have two alleles each, but it is not in all cases. Dominance is always suppressing recessive. It is not in all cases. So there are some traits that do not obey Mendelian law of inheritance. We call them non-Mendelian inheritance or exemptions to Mendelian principle. There are exemptions, right? And these are the incomplete dominance, codominance, and multiple alleles. So what is incomplete dominance? Simply put, incomplete dominance, as the word implies, incomplete dominance. When the dominant allele is not completely dominant over the recessive allele. Don't forget, here we are looking at exemptions to Mendelian principles. Now, Mendel said that when you cross a red flower when you cross homozygotes with two homozygotes, whether homozygous dominant or homozygous recessive, you are going to have heterozygotes, and the heterozygotes will be the same as homozygous phenotype. But in incomplete dominance, where the dominant allele is not completely dominant over recessive allele, you are going to have a blend of the two homozygotes. The two homozygotes are homozygous red and homozygous white. So when you cross a red flower with a white flower, you are going to have a blend of red and white, pink. So that is incomplete dominance deviation from Mendelian law. So incomplete dominance occurs when the heterozygous phenotype is a blend of the two homozygotes. Here, see the heterozygous phenotype, heterozygous RR. It is a blend of red and white. If we were to follow Mendelian principle, this guy is supposed to be red, yes or yes. But here, this inheritance of this color in this plant snapdragon does not follow Mendelian principle. So that is one exemption to Mendelian principle. So if you cross a red snapdragon with a white snapdragon, you get pink, which is a blend of red and white. That is, these intermediates. The heterozygotes are intermediates of the two homozygotes, or a blend of the two homozygotes. Okay, that is incomplete dominance. Now, when you now serve this heterozygotes, you are going to have something that looks like this. So when you now serve F1, when you now serve heterozygotes, you are going to have this Punnett square. Because they are both homozygous, you are going to have these genotypes, these gametes here. When you cross this, you have this. Let me use another color for that. Let me use color blue on red. Yeah, should fit. So when you cross this, you are going to have this, which will be red. This and this will give you heterozygotes, which will be pink. This and this will give you heterozygotes, which will be pink. Then when you cross the two recessives, will give you your white color. So the F1 generation will be all pink, but when you now serve the pink, you're now going to have three different phenotypes: red, pink, and white for F2 generation phenotypically. But you know that if we were to follow Gregor Mendel, we're supposed to have 3:1, because all of these homozygous and heterozygous are supposed to be red if we were to follow Mendelian principle. But this one is non-Mendelian inheritance. Then let's look at another one is codominance. Codominance is when you have three alleles, right? And one is not dominant over the other among them. Example is your ABO blood group, right? Where A and B are codominant. A and B are codominant and O is recessive. So wherever you have A and O, the individual will be A because A is dominant over O. But there are some individuals that are of blood group AB, right? That means A and B are codominant. So when you have A and B together in an individual, the A will not suppress B and B will not suppress A, and they will not produce a blend like you have an incomplete dominance, but rather both of them will be expressed. So heterozygotes have both traits expressed in an individual, right? So this is heterozygotes, A and B. So you will have in this individual blood group AB because A and B are codominant, but O is recessive. So if you have genotype BO, the individual will be B. If you have genotype AO, the individual will be A because A and B are dominant over O. But when you have A and B together, co-dominance will come into play there. Then we have this slide that illustrates further what we can have as a blood group. So an individual can have 6 possible genotypes with 4 possible phenotypes, right? So if you are blood group A, you can either be AA or AO. If you are blood group B, you can either be genotypically blood group BB or BO. If you are blood group AB, genotypically you are AB. And if you are blood group O, genotypically you are OO. So that is a codominance in a what's it called? Codominance as an exemption to Mendelian principle. Then the last exemption is a multiple allelism. Multiple alleles simply means a trait having more than two alleles. You know, Gregor Mendel said that, Gregor Mendel said that for a particular trait, you have two alleles, the dominant allele and the recessive allele, right? But here we are having 3 alleles determining a trait. And we are going to use our example blood group as well. Blood group is a single trait, right? But you have 3 alleles, allele A, allele B, and allele O. And 2 of them can combine to specify the phenotype in an individual. We're not talking about codominance here. So we are using blood group to explain codominance and multiple alleles. Multiple alleles means several alleles. Gregor Mendel said only 2 alleles per trait, but here we are having a blood group that has 3 alleles. 3 alleles. Okay, then another example is seen in a inheritance of coat color in rabbits. In rabbits, there are 4 alleles that determine coat color. But Gregor Mendel said that all traits have what? 2 alleles. Green or allele for green or yellow, allele for tall or short, allele for round or wrinkle, allele for axial or terminal for flower position. But here we are having 4 traits that determine coat color. Coat color is just just one trait in rabbits. But we are having 4 alleles that specify coat color. These are traits that do not obey Mendelian principle. They don't obey what Mendel said about genetics. So that is why the law of dominance is not reckoned with in genetics. Okay, here we have 4 alleles. Let's go very quickly because of those that have 9 o'clock class. We have 4 alleles that each have different phenotypes. Now, let's look at this table. We have a table of 3 columns here, allele, phenotype, and inheritance pattern. The first one is capital letter C, and that's the phenotype for that allele is dark gray. The second allele is small letter C, superscript CH, and the phenotype will be chinchilla. That will be the color now. Then the next allele, the third allele is small c and superscript H, and that phenotype will be Himalayan. And the last allele for coat color in rabbits is a is recessive, a small c, a recessive, which will be white or albino. I hope you are getting it. I'm going very fast because of people that have class, but let me quickly take it again. We said multiple alleles. When you have more than 2 alleles determining a particular trait. In human, we look at ABO blood group. ABO, there are 3 alleles in blood group, allele A, allele B, allele O. Each of them will determine, 2 of them will combine together and determine whether you are blood group O. The phenotype there is a blood group O. Okay, when we do your blood group, what can we see? A, your blood group is blood group O, but you have 3 alleles that can combine different forms to determine your phenotype, your phenotype, your blood group. Now, again, in rabbits, coat color, there are 4, right? Is determined by 4 different alleles, and they can combine different ways to determine what you have, right? So we said there are 4 alleles that determine coat color in rabbits. We have the dominant C, which specifies dark gray coats, the recessive C with superscript CH, which specifies chinchilla, the recessive C with superscript H, which specifies Himalayan, and the recessive C, which specifies white. And what is the inheritance pattern? The inheritance pattern is the order. The dark gray coat is dominant over all the remaining 3. Then chinchilla is dominant over Himalayan and white. Himalayan is dominant over white, whereas white is the recessive. Now, let's look at this example to buttress our point. We said, suppose you cross a chinchilla rabbit with the genotype shown in brackets with a dark gray rabbit with the genotype shown. What are the possible offspring with genotypic and their phenotypic ratios? Now, we have this genotype. This genotype can only have 2 gametes, C superscript CH and C superscript H, like you have here. The gamete for this guy is here. So when you cross, you have this, and this. Now, this guy, this one will be dark gray, because dark gray, capital letter C is here, is dominant. This guy will be dark gray. This guy will be chinchilla. And this guy will be Himalayan. And that is inheritance of coat color in rabbit. We have the slide, and you'll be able to see further in your example. Then I think that should be the last slide. Okay, we still have the one that talks about applications of genetics. Genetics, as found, is placed in different fields in agriculture, to have food security, to introduce traits of interest to a particular plant. Maybe you have a plant that produces a small fruit, but you need a big fruit. You can alter the gene, relying on your knowledge of genetics. In medicine, we now have what we call personalized medicine or pharmacogenomics. You rely on your genome, whereby your treatments is targeted as your own genome. So if I have headache and you have headache, you might be given different drugs to cure our headache. In forensics, in paternity testing, in crime detection, using DNA analysis to know who commits crime. And in evolution, genetics have played significant roles. You may want to form notes on these 4 aspects of application of genetics. And thank you for listening. Do have a wonderful day. I'm sorry for taking your 3 minutes. Do take care, guys. And we may not be able to take any question, but if you are not offering chemistry, which I don't think there's anyone taking biology without chemistry, you may ask your question or send your question to my WhatsApp, and we will take it. Okay, this person has a question. Can a child whose parents are blood group O be blood group A, AB, and O? 100% no to Fatima Teniola. If your parents are blood group O is a recessive trait. So if you are blood group O, it means you are OO. There's no way you can father children with blood group A, B, and B. But if your parents are blood group A or B, then you can be O. But if your parents are both O, there's no way you can be A. Okay, if you need further explanation on that, you can enter my DM and I will reply you. Okay, thank you everybody for joining. Please go for your chemistry class. Do have a wonderful day. So what of people that their blood group is O, but their genotype is AA? Genotype is different from blood group. They are different traits. Genotype is different from a blood group. You can be blood group AA and that. So the homozygous allele cannot make a gamete. I don't know what to mean by that. Homozygous allele cannot make a gamete. Okay, guys. Bye-bye and have a wonderful day. All the best in your test and happy Valentine's Day.